The very first thing that has to happen in order for a virus to infect a host cell is an interaction between a protein on the surface of the virus and a protein on the surface of the cell. That process, that attachment that goes on, gets a name. We call that adsorption. It requires a very particular interaction between two proteins. They have to match each other, they have to fit together. Sometimes those proteins are a little bit unusual in that they may have bits of carbohydrate on them or bits of lipid on them. So of course they would then be referred to as glycoproteins or lipoproteins. But the important thing is that the two proteins fit together, and when they attach to each other, a shape change happens. And that allows the genetic material in the viral particle to penetrate into the cytoplasm of the host cell. Remember, mutations affect adsorption. Mutations can affect whether or not a virus can bind to a cell. Now, when it comes to viral life cycles, what exactly happens from the point of adsorption onwards? There are really two different types of life cycles. There are what we call the lytic cycle and the lysogenic cycle. We're going to use the bacteriophage as our example virus as we talk through these life cycles. So, just like other viruses, bacteriophages can be either lytic or lysogenic. Remember, that word lytic translates as to lyse or to burst. These are the viral life cycles that end up killing the host cell because they rupture the host cell, they break out of the host cell. Lysogenic viruses are different. Lysogenic viruses actually get into a host cell and then stay in that host cell, sometimes for quite a long time. Once they're inside the host cell, lysogenic viruses can reach a point where they will revert to a lytic life cycle, and then they will exit the host cell. Now, there is one family of bacteriophages. They're called Plasmaviridae, that can exit a bacterial cell without killing that cell. But that's only one family. Most bacteriophages will ultimately kill their bacterial host cell, whether they do it immediately, the lytic life cycle, or whether they do it after some period of time, the lysogenic to lytic life cycle. Eukaryotic viruses are very different. Eukaryotic viruses, as you might expect, have a wider range of life cycle options. Yes, there are lytic eukaryotic viruses, and there are lysogenic eukaryotic viruses. But the way that eukaryotic viruses exit host cells has a little more nuance in it. There are eukaryotic viruses that can bud out of a cell. Remember, we said a lot of animal viruses are enveloped. They bud out of the cell and they take a little piece of the cell membrane with them. Sometimes eukaryotic viruses actually use a host cell process called exocytosis to leave the host cell. And again, that doesn't kill the host cell. So eukaryotic viruses, they have a wider range of life cycle varieties, if you will. They are more complex in the processes that they use. We're not going to focus on eukaryotic viruses as we discuss life cycles. We're going to stay focused on bacteriophages as we discuss these life cycles. Lysogenic bacteriophages are going to establish a long-term relationship with the host cell. What a lysogenic bacteriophage can do is get its genetic material inserted into the host cell's genome. Viral genes, in other words, are going to be passed down from parent cell to daughter cell, from generation to generation to generation, as the bacterial cell passes its own genes from generation to generation to generation. Remember, the bacterial cell can't tell viral DNA from its own DNA. So as the viral DNA sits inside the chromosome, the bacterial cell just keeps doing what it does with its own DNA. Viral genes will also get expressed as bacterial genes get expressed. They'll be transcribed and they'll be translated, and you'll get viral protein production. And as long as that viral DNA stays inside the host cell's DNA, those viral proteins are sometimes used by the host cell. They find them helpful and useful, and they give that host cell new traits, at least as long as that viral DNA remains inside the host DNA. This is a complicated flow chart, but it represents for us the pathway of a lysogenic bacteriophage and how lysogenic phages can revert into the lytic life cycle. So it has for us both life cycles in one drawing, the lytic and the lysogenic. Let's walk through it together. At the top of the slide, you can see a representation of a bacterial cell. And this bacterial cell has its own DNA, and they've shown that to us as this green bar. Now, along comes a bacteriophage, and they're showing us that in orange. And that bacteriophage is going to infect this bacterial cell. So here is the viral genetic material, and that's shown in dark green. Now, if this is a lytic bacteriophage, what's going to happen next is this pathway over to the left. The injection of the bacteriophage's genetic material actually causes the bacterial cell's genome to fragment. Now that sounds terrible, but it doesn't kill the cell. It just means that that circular chromosome gets broken up into pieces. That way, the viral DNA and the bacterial DNA are kind of similar in size. So we have lots of fragmented DNA in here. Some of it is viral, some of it is bacterial. It still functions, it'll still get replicated, it will still be expressed. It's just that it's all broken up into pieces. So the host cell, the bacterial cell, is going to copy viral DNA. It's going to express viral DNA right along with its own. It's going to make viral proteins. And all that new viral DNA and those new viral proteins will then assemble into new particles inside the host cell. Remember, a lot of viral proteins self-assemble. So the host cell doesn't need to do anything really for these new nucleocapsids to be formed. They form spontaneously. Once all of these new viral particles are in place, the lytic event happens, the lysis happens, and the particles burst out of the host cell. They break or rupture the host cell, and they are released. So we started off with one viral particle up here or up here, and now we have many viral particles, and these are all going to go out and infect new bacterial cells. Now let's take a look at the right side of the diagram. So again, we have the bacteriophage infecting that original host cell, injecting its DNA, and now we're going to go into the lysogenic pathway. Now we're dealing with a bacteriophage that's capable of a lysogenic life cycle. So in this case, the viral DNA is going to be inserted into the DNA of the host cell. There's a term on this slide. We haven't talked about the term prophage. Don't worry about that. Just understand that now inside the chromosome of this bacterial cell, there's a stretch of DNA that doesn't belong to the bacterial cell. It belongs to the virus. But the bacterial cell doesn't mind. It can't tell. So it's just going to go on and live its life the way it always has. It's going to replicate its own DNA every time it divides, and it's going to express its DNA. It's going to do transcription and translation and produce proteins that it needs each and every day. Along the way, it's also going to transcribe and translate the viral DNA and make some viral proteins, and it may find those viral proteins quite useful. Life will go on this way until that bacterial cell experiences some kind of stress, some kind of event where it is under stress. We don't really understand all of these signaling pathways, but something is going to put the survival of that bacterial cell in jeopardy. It might be not enough water. Remember, we refer to that as desiccation. It's one of the worst things that can happen to a bacterial cell. It can trigger sporulation in bacteria that are capable of sporulating. So drying out, lack of water, or perhaps a lack of nutrients, or perhaps a change in environmental temperature or environmental pH. Something is going to stress that host cell. And that stress event triggers this viral DNA to exit out of the host cell chromosome and send us back into the lytic pathway. Once that viral DNA is excised or removed from the host cell chromosome, now we're going to see the lytic pathway induced. The viral DNA will be replicated, it will be expressed, new viral particles will be produced, and then there'll be a lysis event, and the bacteriophages will move on to infect other cells. So while we haven't talked about eukaryotic cells in any detail, just remember that eukaryotic cells have a wider variety of exit options when it comes to leaving a host cell. Yes, there are lytic eukaryotic viruses, there are lysogenic eukaryotic viruses, but there are also eukaryotic viruses that bud out of host cells. Some of the herpes viruses do that. The rabies virus does that. Measles and mumps viruses do that. And there are some eukaryotic viruses that take advantage of a normal cellular process called exocytosis to get out of the host cell. In both of those cases, in the budding case and the exocytosis case, the host cell is going to survive that. There's no lysis of the host cell. It's lysis that kills a host cell. In comparison, budding, which takes a piece of membrane with it, that's how an enveloped virus is created, and exocytosis, which is just a normal cellular process. These two types of exit strategies don't kill the host cell, but of course lysis does.